All right, hello everyone. My name is Nick, and I am going to do a little presentation on beta-lactam antibiotics. So here we go. Okay, so first, kind of an overview about where are we going. So we're going to kind of look at what makes bacteria vulnerable. So starting with why are bacteria susceptible to these antibiotics. Then we're going to talk about what specifically is a beta-lactam antibiotic. How do these specific antibiotics work? What are resistance mechanisms that bacteria have developed to combat these antibiotics, and kind of like now what? So, what do we do now that we have a lot of resistant strains, and how do we envision this in the industry of pharmacology? Yeah. So, first, why are bacteria vulnerable to this type of antibiotic? So, both gram positive and gram negative bacteria have what's called a peptidoglycan layer in their cell wall. And this layer utilizes several different enzymes, including transpeptidase and transglycosylase. And so the goal is to cross link these peptidoglycans in order to create strength and to create more stability within the cell wall of the bacteria. So if we look here, the goal is this penicillin binding protein, okay, which are referenced as PBPs. And so this would be a transpeptidase. The goal is for it to interact with this peptidoglycan and link these peptidoglycans together in order to create more strength and stability. And what happens is beta lactam antibiotics. So, if we take something like penicillin, so here, this penicillin, what they do is they inhibit this cross linking process, which ultimately makes the cell wall of these bacteria weaker. So, cross linking of this peptidoglycan layer. So, this is what gives the bacterial cell wall strength. And so, if that strength of the cell wall is interrupted, it makes the bacteria susceptible to lysing. So, what we end up with is ultimately this peptidoglycan layer needs to be cross linked. And what we have is this transpeptidase, the PBP enzyme, wants to interact with this D-alanine position and to link these two together. And I want to focus in on kind of what is happening with this transpeptidase here, because this is why beta-lactam antibiotics function to interrupt this process. So this is just kind of giving us a little bit of an overview of the interaction of the enzymes. And what we're getting is the linking of these two neighboring peptides. So this cross-linking functions through an acylation-deacylation process. And what happens is in the final assembly of this cell wall, we have the D-ala terminus of the peptide removed. So if we look here at this position, what we are getting is the attachment of these neighboring amino group, resulting in an acyl species that is cross-linked to this neighboring chain. So when these two peptidoglycans are linked right in this position, we see more strength within the bacterial cell wall. So the goal of the beta lactam antibiotic is to interrupt this process. So if we look here, I think this is like a really good position to look where this cursor is. This is going to be exceptionally similar when we analyze the beta-lactam antibiotic. So in this position, we've got acylation happening by the penicillin binding protein, and then we get linking when this when these when this all right, let's pause this, cut this section. Okay, pause, cut this section. So if we look here, we get cut. Okay, so let's take a look at this image from Fisher. So I want to kind of look closely at this position because this is exceptionally similar to penicillin. Okay, so we're going to kind of think about this position here. So what we end up seeing is that in the last step of cell wall assembly, this D-ala terminus is removed and the resulting acyl species is cross-linked to an amino group of the neighboring chain. So here we've got this neighboring chain and we have cross-linking occurring, which creates more strength in the cell wall. And so penicillin is going to mimic this termini, which makes it recognizable by transpeptidases. So instead of engaging in this process of acylating and deacylation to cross-link these two strands together, penicillin inhibits that process. So let's take a look at what do these molecules look like and how can we think about these similarities. So a beta-lactam antibiotic has a cyclic amide attached to the beta carbon. And originally, this was limited to penicillins and cephalosporins, but now it's a bit more expanded and it includes different synthetic monocyclic beta-lactams, carbapenems, oxapenems, carbacephams, and oxacephams. And I'll include a structural image on the next slide if you want to take a look at some of these, but we're mainly going to focus on penicillin's interaction here. So again, the goal is for these antibiotics to target the transpeptidase enzyme, disrupting cross-linking of this peptidoglycan layer in the bacterial cell wall. Okay, so here are some of these structural beta-lactams to see kind of what these structures look like. And yeah, mainly we're going to focus on penicillins here, or I'm going to focus on penicillins here. But if just for some reference, here are some other clinically relevant beta-lactams. So ultimately, the enzyme interacts with penicillin instead of peptidoglycan at the D-ala terminus. So they disrupt this cross-linking because they're causing interaction with the penicillin binding proteins instead of the cross-linking occurring. So in what we would normally see in the cross-linking of these strands here, we have this transpeptidase enzyme interacting with this D-ala terminus, ultimately cross-linking these two peptidoglycan strands. But in the presence of penicillin, okay, so we've got, this is our molecule of penicillin here, which mimics this terminus very closely, which causes the transpeptidase enzyme to interact here instead of with the peptidoglycan. What this does is it occupies the enzyme's active site, renders it unable to cross-link the peptidoglycan, leads us to extremely weak cell walls, which will lyse with very small changes in osmotic pressure. This process causes inhibition because the hydrolysis occurs at such a slow rate. So the hydrolysis of this process is occurring so much slower than, or it's occurring at a much slower rate than the bacterial replication. So they're going to lyse much more quickly than they will lyse with the small changes, which won't allow them to replicate and ultimately lead to their death. Okay, so what are different resistance mechanisms that have developed within bacteria that are resistant to these types of antibiotics? So mainly there are two modes of resistance that occur. One is a mutation in the penicillin binding protein, which causes changes to the transpeptidase enzymes. And another mode is what are called beta-lactamases. And so these are enzymes that are ultimately causing destruction to the beta-lactam ring. So we got 4 different classes of these, A, B, C, and D. Okay, So let's look at the first mode, which is PBP mutations. So I'm going to look at a specific example. So we can look at Staphylococcus aureus here. And staph has acquired resistance through the mode of a penicillin binding protein mutation. So, in susceptible strains of Staphylococcus aureus, there are 4 different transpeptidase enzymes that have differences in their molecular masses. So, what we see are 3 larger enzymes and 1 smaller enzyme, and these are affected or inhibited by beta-lactam antibiotics. But when we look at resistant strains have 5 of these transpeptidase enzymes. So in the resistant strains, they've got 1 extra enzyme, which has been termed the PB2A. And this enzyme is a low affinity enzyme to beta-lactam antibiotics. So therefore, while other PBP enzymes are unable to cross-link, the PB2A enzyme continues to carry out its enzymatic activity and is able to cross-link the peptidoglycan layer. So this is one mode. Conformational changes to these enzymes rendering them able to continue to carry out cross-linking, which would lead to a functioning cell wall. Okay, the second mode is what are called beta-lactamases. So these were identified prior to large-scale penicillin production, and there are 2 major types of these. So the first major type is those that use serine acylation, deacylation, and these are termed classes A, C, and D. They would be the serine-dependent enzyme classes. Then we also have a metal-dependent enzyme class, which mainly utilizes a zinc ion. So the goal of these beta-lactamases is to deactivate the beta-lactam ring. So this is going to be accomplished through hydrolytic deactivation. So an acylation, deacylation of this ring here. When the ring is intact, the transpeptidase enzyme is going to interact with this molecule of penicillin, which is going to leave it unable to cross-link. So it's going to interact with this penicillin molecule instead of interacting with the peptidoglycan. So when there is a beta-lactamase present, like penicillinase, what happens is the goal is to drive the opening of this lactam ring. So through acylation, deacylation, if we can open this ring, this now renders the penicillin inactive. So it can no longer be an inhibitor for the transpeptidase enzyme. So once this has been achieved, if it can't interact with the PBPs, then the cross-linking is going to carry out and the bacteria will be able to survive. Okay, so kind of some questions, just like problems is what are the problems here and where do we go now? So the big problem is that the widespread use of beta-lactam antibiotics, penicillins, and cephalosporins has ultimately led to antibiotic-resistant enzymes that survive and reproduce because we've basically knocked out all the bacterial strains susceptible and created unlimited resources for the resistant bacteria to survive and pass genes on to the next generation. Canaliva argues that we have to do more than simply look at disrupting peptidoglycan linkages for antibiotics. And ultimately, what we have to do is learn to target other non-traditional molecular targets in order to increase the efficiency. So there are several different possibilities for this in order to increase efficiency of antibacterials. But kind of the idea that, hey, let's move beyond the peptidoglycan linkages and these cell walls and think about other aspects of the bacteria that we could render ineffective. So thanks, and please let me know what questions you have.